Good morning, everybody. My name is David Ramirez, and today I would like to talk to you about the work we have been doing in collaboration with people from the University of Copenhagen in Denmark and people from University of Talca here in Chile. The name of this poster is Structural Insight of GTRs, Substrate Specificity and Transport Mechanisms through Fast Dynamic Docking guided by Adaptive Electrostatic Bias. GTRs are glucosinolate transporters. So these guys are the GTRs in the inward facing conformation, as you can see here, and the substrates are these glucosinolates. We are studying these two different glucosinolates, I3M and 4-MTB. I3M is derived from tryptophan, that's why the side chain is an indole, and 4-MTB is from methionine, that's why the side chain is an aliphatic side chain. So these are the only difference between both substrates. The rest is the backbone is the same. This glucose and the sulfate. So the main motivation for us is to study how these substrates actually reach the binding site. Here in 2017 we published this paper when we discussed how different glucosinolate transporters are, in this case GTR 1 and 3, are able to transport with the same activity both substrates, but GTR 3 somehow is just able to transport I3M but not 4-MTB, as we can see here. So the main question is why GTR 1 transport both substrates whereas GTR 3 only transport indole glucosinolates, in this case I3M, the green one. And both GTR 1 and 3 shares around 60% of sequence identity. So that's why it's a very interesting question for us. How we tackle this question? We built different systems using GTR 3 in inward facing conformation. That's why here it's saying in wild type with both substrates I3M and 4-MTB. And as a positive control, we use this peptide transporter, the name is GK pod, in the inward facing conformation, and in this case is this transporter is crystallized with alaphosphalline, which is the substrate. So we know which one is the crystal structure, which one is the binding site of this, or which one is the binding site of this alaphosphalline. So that's why we use this structure as a positive control. Here you can see the PDB code. So why only inward facing conformation? Because both models were modeled using the nitrate peptide transporter NTR 1.1, which is crystallized in the inward facing conformation. And this one is the only available crystal structure that we have so far. How we tackle this question? We construct 50 replicas using 20 nanoseconds per replica, or per system actually. So at the end of the day, we run around 3 microseconds of molecular dynamic simulation, because we have 3 systems for GTR3 with I3M, GTR3 with 4-MTB, and the GK pod with alaphosphalline. And we run this fast dynamic docking using Vicky, or molecular dynamic docking using Vicky, which employed an adaptive electrostatic bias. So we apply a force and we pull in the substrate to reach the binding site, and then we release the substrate, and regular classical molecular dynamic simulation is performed to reach the final time, which is 20 nanoseconds. So here you can see how actually the initial position is. The substrate is in the intracellular space. It's not interacting with the protein, and then we can see here how we pull in, applying this force, we pull in the substrate, and around the 6 or 7 nanosecond we release the force, and we run classical molecular dynamic simulation, as you can see here. So because we are using a force, we are using an electrostatic bias, so there is a work So we calculate the work for all the replicas, we normalize the work, and then we just select the best or the replicas with the lower normalized work, which is under the 25%. Those replicas, in this case for GTR3 wild type with 4 MTB, we found 27 replicas to be analyzed. And this video is just for the replica number 1. So at the same time, we calculate the work, and at the same time we calculated the RMSD against the RMSD of the substrate atoms, and as a reference we use the initial position, which is outside of the, in the intracellular conformation, right? So that's why the RMSD is very, very big. The first around 6 nanoseconds is with this force, so that's part of the MD is a non-equilibrium molecular dynamic simulation, and then we release the force as the substrate remains within the binding site and is somehow moving around, but is accommodating there, and that's what we mention as an equilibrium molecular dynamic simulation. And we can see that after around 6 nanoseconds, the RMSD of the 50 replicas, this is the average between all of the replicas for the system, is more or less the same. So here we can see here in the left part the normalized work for the 3 systems, and if we put this line here in the 25%, we can see that for GTR3 with 4 MTB, we found 27 replicas. With GTR3 with I3N, 26 replicas. For the positive control, 23 replicas. So it's more or less the same, more or less 25 replicas per system. And here we can see how the substrate atomistic RMSD change. So we have here the 3 systems, and then we also compare how the JK pot wild type with alaphos, how the alaphosphalline atoms are changed, but as a reference we didn't use the initial conformation, we use the crystal structure, so that means the final conformation of this alaphosphalline. And we can see here that the RMSD is around 4 Armstrong, which is really good actually. That means that this protocol is able to reach somehow the binding site that is described by the crystal structure. Then, using just the top 25% of the replicas, in this case I am just showing you guys the result for GTR3 with wild type, but we did the same for all the systems. So here we can see the trajectory and the center of mass of 4 MTB, and how is the trajectory. We clusterize this in the entire replicas. You can see here different clusters. Now we have like significant clusters. That means that now we are able to study how these different conformation along the pathway are interacting with this, in this case with this receptor. And in the equilibrium molecular dynamic simulation, we analyze all replicas. That's why we have more clusters, and we select the significant clusters here. So we merge all the replicas and we clusterize using GROMACS and 2 Armstrong threshold. And at the end of the day, we got 3 different clusters that you can see here with different populations. So now the task we are facing is to study how these clusters are interacting along the pathway and also in the central binding site with the substrate, with the transporter. We also analyze using the Jarzynski equality for non-equilibrium MDs. We analyze how the binding energy is changing along the simulation. So if we have the work, we actually are able to calculate this delta G. So the delta G was lower for the system with the I3M in comparison with the system with the 4MTB. That makes sense because the experimental data that we have say that GTR3 is able only to transport I3M, I mean in an active way in comparison with 4MTB. So these results actually is validated by our experimental work. So now we are studying how the substrates are interacting with this transporter along the pathway binding sites. Hope you enjoy, and if you have any question, feel free to just to type some of the question, and I will be happy to discuss more about our work and our setup. This is an progress work, and we hope to finish the experiments actually, because we need to validate, we need to find key residues and then go to the lab and mutate those residues to see if actually we are identifying like molecular determinants in the substrate and transport mechanisms of GTRs. Thank you very much for your attention.